Hi, in this video we'll talk about the cell cycle, which describes the various stages a cell has to go through to divide and reproduce itself. We'll look at two viewpoints of the way of looking at the cell cycle. One is the modern or molecular-based view, started to appear in the 1980s, and it has four stages: a DNA synthesis stage, a mitosis stage, and in between there are two G stages. These can stand for gap or growth. So there's growth 1, synthesis, growth 2, mitosis. Prior to the 1980s, the tool that one used is microscopy, and that gives a very different picture of the cell cycle. That is based by the acronym IPMAT for interphase, prophase, metaphase, anaphase, and telophase. And then we'll try to make a correlation between the two and see the different emphases that the different tools that we use produced on the molecular events. We'll talk about checkpoints. These are quality control points that the cell must satisfy before they can transition between the different phases. And then we'll talk a little bit about the molecular machinery, proteins named cyclins that regulate the transition for new phases if the cell satisfies the requirements of the various checkpoints. On this slide we see fluorescence micrographs of cell dividing. Now the cell exists in this phase, the interphase, for about 95% of the cell cycle. The interphase also corresponds to the G1, S, and G2 phase. These other stages here are the mitosis stage. And as you can see, you have the condensation of chromosomes, them lining up in metaphase, them pulled apart to each of the daughter cells, to different poles, the cytokinesis occurring, and finally two identical daughter cells. And on this slide, you see the cell cycle in its most basic. Here we have the starting cell. The first job is for it to duplicate the chromosomes. Then the chromosomes are segregated to different ends, and then the cell divides. This is a cell cycle in its very basic configuration, and cells do these over and over again. Now this is a depiction of the modern view of the cell cycle. Again, we have the G1, the S, and the G2 phase. This is going to correspond to interphase, and this is greater than 95% of the time of the cell cycle. The M phase can take about an hour for eukaryotic cells, during which you're going to have mitosis, nuclear division, and cytokinesis, cytoplasmic division. You can refer to, as many biologists do, mitosis to represent both of these processes, but technically cytokinesis represents the cleavage of the cytoplasm. Lastly, at this point, I'll just say that the S phase, this is the critical phase where your chromosomes are replicated in preparation of passing on a set of chromosomes to both daughter cells. Now here is the visual-based view of the cell cycle. And we see the long process of interphase. And when I studied biology in the 80s, we basically didn't even cover it. We just mentioned that it existed between all these interesting phases. Even the name interphase, like intermission, is just something that has to happen every once in a while, revealing the bias of earlier scientists. But here again we see the condensation of chromosomes into their compact forms, lining up in the center of the cell, and then separation of the chromosomes, the formation of two new nuclei, and the formation of two new cells. We'll go through each of these stages: prophase, prometaphase, metaphase, anaphase, and telophase in detail in a future video. However, I think that you should memorize now metaphase. This is sort of the middle of the phases. It's when all the chromosomes line up in the middle. And then if you remember the mnemonic IPMAT, interphase, prophase, prometaphase, and you're trying to remember what molecular events occur during them, you'll remember that these are the things that have to be done in preparation to get the chromosomes lined up. And then if you think about anaphase and telophase, you'll know that these are things that come after metaphase, and so you'll be able to remember better what goes on here. But we'll see this in more detail soon. And again, here we go putting the two views together. During interphase, we have G1, S, and G2. So if you wanted an all-purpose mnemonic, this would incorporate both of the views, but that's probably not super helpful, other than to make it clear that regardless of the viewpoint that we take, we're talking about the same set of events. This slide just shows some typical cell cycle times. This is only for eukaryotes. For bacteria, E. coli is about 40 minutes. Early embryos, eukaryotic embryos, can be about that time scale. Yeast cells, one of the simplest eukaryotes, can vary between 90 minutes and 3 hours. Liver cells in your body divide about once a year. If you do experiments in labs with culture, cells divide about once a day. In a few places in your body, in the intestine where you have a lot of cell turnover, or with your immune system, they can cycle in less than a day. Now in this slide we're emphasizing the role of checkpoints in the cell cycle, and as I mentioned, these represent attempts to quality control the state of the cell before proceeding on to the next stage. Now in slowly dividing cells, or stagnant cells, they're hanging out either in G1 or a closely related phase, G0, which we'll talk about. And the major checkpoint is the start checkpoint. This is where if a cell decides to divide, this is the committed point to completing a cell cycle. So cells will generally only cross this checkpoint if the environment is favorable, both from a nutrient standpoint and from a signaling standpoint. Do we have the right growth factors signaling us to proceed in the cell cycle? If the cell senses that the environment is not favorable, it will stay here in the G1 phase and wait until the environment changes. But if these things check out, it will enter into the S phase. It will begin the process of duplicating the DNA, the quality of which is being monitored all the time. But then we'll, in terms of checkpoints, we'll talk about the G2M checkpoint. And this is after synthesis and growth, before we commit to cell division, before we enter in mitosis. You want to make sure that all the DNA is replicated, that any damage has been repaired, any mutations have been repaired, because once the cell divides, there's no going back. If the cell determines that the DNA has been replicated properly and is ready to go, it will enter into mitosis, and things will be lining up into metaphase, where the chromosomes are getting ready to separate. But before they proceed to anaphase, before they start pulling the chromosomes apart, there is a checkpoint which wants to make sure that the chromosomes are properly attached to each side and that each daughter cell will receive a full complement of chromosomes. The way the cell does this is a beautifully mechanical mechanism, very logical, and we'll be speaking about that in more detail in a future video. This cell cycle timeline indicates a few additional checkpoints, but again, we're going to talk about 3 of them for the purposes of this class. The start checkpoint, the commitment to enter into the cell cycle; the G2M checkpoint, the commitment to enter into mitosis; and then the metaphase to anaphase transition, the dispensation of a set of chromosomes to each daughter cell. On this slide, we see the first view of the protein machinery which governs the transition of cell cycles stages, and we're focusing here on the transition between G2 or interphase into mitosis, and this is regulated by the M cyclin and the M cyclin-dependent kinase. There are other cyclins and other cyclin-dependent kinases that promote entry into the different phases, but the M cyclin will gradually accumulate inside the cell. When it reaches a certain threshold level, then it will activate the cyclin-dependent kinase, and this kinase will phosphorylate a number of target proteins which will start the transcriptional machinery needed for all the proteins to be made required by mitosis. So the cyclin is a regulator, and the cyclin-dependent kinase is the effector protein for stimulating this transition. Towards the end of the mitosis, the cyclin is degraded. That will drop the activity of the cyclin-dependent kinase, and then the process will start over again where the cyclin is being synthesized, and eventually it will reach the threshold level once again. Now in this slide we see a little bit of the molecular mechanisms of the activation of the cyclin CDK complex. And although the details here are a little bit more than you need to know, the CDK subunit itself is phosphorylated before it's activated. See here, it's not just the binding of cyclin to CDK that activates it. Initially it's phosphorylated, but it's not active because there are also inhibitory phosphates which then get cleaved off, and that produces the final active form. Now this complex with the kinase subunit will go on to phosphorylate transcription factors and other genes that are required for promoting all the molecular events of a new phase. Now in this slide we see that there are multiple cyclins as mentioned. The G1S cyclin, which will rise and then fall, the S cyclin, which will persist for a long time, and then the M cyclin, which appears and then is actually degraded halfway through mitosis during the metaphase to anaphase transition. The M cyclin is degraded, in fact, by the anaphase promoting complex, which is a protease which will degrade the cyclin. Now, I'll emphasize that the CDK portions of the subunits persist at relatively constant levels throughout the cell cycle. It's only the cyclin subunit that's rising and falling in concert with the stage of the cell cycle that it's in. And specifically that the degradation of M cyclin is required for exit of the mitosis phase. I don't believe that the transitions, for instance, from these other stages are strictly dependent on the degradation of the cyclin, but for metaphase it is true that if this cyclin persists, you will never get past the metaphase to anaphase transition. This slide emphasizes again that it's the cyclin that gets degraded. The kiss of death, or the mark for protein destruction, as you'll remember, is polyubiquitination. This targets this protein for destruction by the proteasome. Without the cyclin subunit bound, the cyclin-dependent kinase gets dephosphorylated and exists in an active phase until cyclin builds up again. I'd now like to add two details to the checkpoint story which we've talked about. Now that you know that CDK proteins are the ones that promote the transitions between the different phases, we learned that there are specific CDK inhibitor proteins which will, if the cell does not satisfy the checkpoints, these inhibitor proteins bind to the CDK, and so that you're not going to get transition to the next phase. And then I'd also like to specifically call out the G0 phase, which is sort of a parking lot, which we'll see, where cells, as they're dividing, like the liver cells, which take a year to divide, most of the time they're sitting here in this parking lot. When they decide to re-enter the cell cycle and consider dividing, they'll transition back into G1, and they will then, if things are favorable, commit to another round of cell cycle. So here's a cartoon of the G0 parking lot where cells will hang out, not only cells that are cycling and taking a long time to do that, but also cells which have withdrawn entirely from the cell cycle, such as the fact that neurons are no longer dividing in the adult brain. They exist permanently in the G0 state. But for cells that are cycling, if the conditions are favorable, they will pass the G1S checkpoint and commit to the cell cycle, which will then take at most 12 to 24 hours for it to complete. G8. On the last slide of this video, I want to talk about one of the DNA damage checkpoints and how it operates. This one is the best studied, is the one that occurs in G1, so it's not the one that we mentioned or emphasized at the G2 to M transition, but the basic principles are largely similar. The key protein in the DNA damage checkpoint is p53. This protein is known as the guardian of the genome. It's the most frequently mutated protein in all cancers. Now, p53 is constantly being synthesized, but it's also constantly being degraded. So in the absence of DNA damage, p53 is maintained at low levels. So synthesized, degraded, synthesized, degraded, synthesized, degraded. If DNA damage occurs, proteins bind to the damaged DNA and will also phosphorylate p53 and making it stable. So it's going to block the degradation of p53. It keeps getting made quickly, so the levels of p53 will quickly rise. Activated p53 is a transcription factor. It's going to, among other things, transcribe the p21 gene, and p21 is a specific inhibitor of the G1S cyclin-dependent kinase. So this is going to block entry into S phase as long as p21 is bound to the CDK. Now that the checkpoint has been blocked, the cell has to make a decision: cell cycle arrest plus DNA repair, or give up, commit cellular suicide known as apoptosis. And the mechanism for this decision is pretty interesting, or the suspected mechanism is. It depends specifically on the amount of DNA damage. The more DNA damage you have, the greater the amounts of p53 that will occur. At lower levels, as it's starting to accumulate, p53 will bind to only its highest affinity transcription DNA binding sites, and those are the proteins that initiate cell cycle arrest. At very high levels, it'll bind to the high affinity sites and also to lower affinity sites, which will promote apoptosis. So if there's a small amount of DNA damage, the cell will try to make a repair. If it feels like it's overwhelmed with the amount of DNA damage, there's a built-in mechanism based on simple thermodynamics, amounts of p53 and binding affinities at different genes, that will automatically say we give up on DNA repair and we'll just go to cellular suicide. Because the one thing the cell doesn't want to do, almost more than anything, is go ahead and divide if the genome is damaged. Okay, thank you very much.